The Science of Feeding Tomorrow. Pathway Intermediates.
Chromium is this forgotten mineral, and basically, how can it improve aquaculture? Well, let us begin this concept. Fish nutrition. Well, our understanding of macronutrition for protein and essential amino acid requirements is well known for fish and shrimp as well. We must not forget the shrimp, but the fish, of course, we know much more about fish. The protein to energy ratios have been established. We know a lot about the lipids, and the essential fatty acids have been defined largely for most species, but we still have lots to learn. The vitamins are also an important group of micronutrients, and I've been working in the past on various types of vitamins, including the B vitamins and the fat-soluble vitamins, and especially vitamin E. But the mineral nutrition, well, we could say, I suppose, that there's still a paucity, a gap of information for lots of cultured fish, especially marine species. And so because the marine environment is relatively new in aquaculture, we farm mainly freshwater species. People are surprised to learn about that. But we need to look at the marine species as well, but there's a lot of trace elements in the marine environment, in the water. But we do need a dietary supply. The current issue in feed nutrition, let's have a look at some of the major areas of interest because they do relate to our trace element story. We have a limited availability now of marine resources. People are cutting back on the use of marine ingredients, and fish meal and other marine sources contain a lot of trace elements. And if we are going to reduce our source of marine supply, we are going to put more dependency on plants and other components. So we are formulating new diets and changing the recipe. So that's going to put some impact on our trace element requirements and specifications. We cannot use in some parts of the world, although it's quite all right to use poultry proteins from poultry as byproducts. But in many countries, like in Europe, we have limitations on how far we can take animal byproducts, which are also very rich sources of trace elements. We have to consider the safety of farm animals, fish, and how it relates to the humans. And we have to be very careful about heavy metal contaminations of different types of feeds. So we have to bear in mind that some sources may contain, high levels of contaminated heavy metals. So we have to be very careful. But we need to maximize also, minimize, I should say, the potential negative impacts of aquaculture on the environment. We don't want to excrete out too much trace elements. Not only are they expensive in the diet formulations, but we don't want to waste them and impact on the environment. For instance, like copper and other metals, which can be an issue. We want to reduce, therefore, the fecal and urinary excretion of minerals and elevate the bioavailability. This is a word we need to use more, the bioavailability and the retention of trace elements in the growing animal. And we see deficiencies. There's a book chapter I wrote which concentrates on nutritional deficiencies, and many of them can lead to bone deformities and aberrations and morphological irregularities in the fish. So we need to look more at nutritional diseases, and trace elements play an important role here. And therefore we need to also define how trace elements affect the immune response of fish, to boost the robustness of the fish and to minimize disease prevention. And also the fish welfare issues related to the fish quality and the taste and texture for the consumer. We know selenium is important in that area of concern. So the essential minerals in fish production, this is a nice overview. You can see here the essentiality has been confirmed by biochemical mechanisms for minerals like macroelements, calcium, magnesium, sodium. These are large electrolytes, which are very important in maintaining also a regulatory balance in fish. And the microelements like iron, zinc, copper, cobalt, like manganese and selenium and iodine and molybdenum are some of a few of these elements. We know a lot about them, and very, very important as microelements and confirmed. But then there are these other areas which up to now have been a bit grey. These are the essentialities suggested by physiological impairment. And there are some very odd and strange ones here. I wouldn't recommend we take arsenic, but you never know that some very, very micromolar levels of arsenic and other elements like fluorine, lithium, vanadium, lead could be important. We think that silicon could be very interesting. But chromium, which I've highlighted there in green, is the essence of our story today. Chromium is definitely one we know has an essentiality linked to physiology and metabolism in other animal species. And we've learned now that it's becoming more and more important and acceptable in aquaculture nutrition. So let me just remind you that the whole body status is very much concerned about balance. It's all about balance. Deficiency here and maybe some uptake from the water, especially in marine systems. And then we get to this point here about balance. At the fulcrum, we have optimization. And I would say that we should be looking at homeostasis, how the fish is regulating, and the shrimp are regulating their supply of minerals. And if we go to excess, well, obviously that's not good. That's toxic up here. But we never get up here very often. We are down here around this area, okay, of homeostasis. And we need to look at this system much more carefully. And here is a nice idealized scheme where we see death, we don't want that, and we have optimal health. We want to be here. We want to be at this zone. Dietary requirement may be shown by enhanced growth, FCR, feed conversion. All these things might be affected, and we may have a requirement here. But the optimum level might be higher because of stress, and because of environmental interactions. And so we want to be at least meeting diet requirements and preferably affecting positively the animal's metabolic status. We never want to be up here. Okay, and this shows it very nicely. The red bar is our known requirements from dietary research from the NRC, National Research Council database for fish and shrimp. Okay, but these are fundamental basic requirements of what we know from the scientific papers. But the blue bars suggest that this is the range. This is the range that we see for research papers in different species, across the species, okay, including shrimps. But very little is known about those in marine environments such as shrimp in high salinity. But the chromium is highlighted here in green. We know that there is this range of requirements from the papers we know so far, but we need to confirm this. So it's around this area of 1 milligram per kilo, but we should be concerned about the ranges as well. And there are many factors influencing the mineral bioavailability, and that is the chemical form of the metal, the trace element, the speciation, the species of it is the term we use. The particle size, the grinding size, the milling properties. We're talking in microns. To add to the feed additives into the premixes, we have to be sure that the trace element is mixing properly. Then we have to be concerned about the water chemistry. How much of that mineral is present in the water? How much is not? How much is required in the diet? And then there's the fish species. How does the fish species relate to this? Some species are more tolerant than others. Some fish will be more stringent. And then the status of the fish, the pathological status or the stress. So I must bring in stress here. Stress situation. Sometimes we are all stressed and we have higher demands for energy, such as before exams in high school, things like that. We're stressed. So the pathological status, when the animal is diseased, they will have a higher requirement to repair the tissues. So the physiological development, the stage of growth, and the reproduction state of the animal or the fish will be especially interesting for us, especially in fish. The digestibility of the diet overall will affect the nutrient supply and bioavailability. And of course, the interaction of nutrients, such as chromium, with other elements, and the interactions with the gut and tissue metabolism will have a play. And then, of course, there may be dietary inhibitors anti-nutritional factors, which can chelate. It's very, very true in phosphorus, for example. We can see this in phosphorus in such compounds as phytic acid, which locks up and binds the phosphorus. Fortunately for us, I don't think there's too many problems with the chromium in terms of that, specific binders, inhibitors. And in minerals and fish nutrition, so we have this very nice profile here. We can see requirements and toxicities in this zone here. Then we see the interactions, mineral and mineral, mineral and protein and amino acid interactions, mineral and lipid and other interactions that we may not know about. Well, ANFs, anti-nutritional factors. And these all affect the bioavailability and excretion. The mineral bioavailability must be high. We're trying to aim for high from the ingredients themselves, where some elements are present in the ingredients, but we cannot rely on that. That is why we need to supplement and add these elements to the diet. We cannot trust the ingredients to supply those trace elements. People often ask me, oh, but surely the minerals are in the feed ingredients. Yes, they are there, but we are manipulating the diet formulations to the point where we need a premix of vitamins and minerals together or separately. So chromium, this is the heart of our story today. Chromium, therefore, I would say, is an essential trace element from our studies with other animal species and now with fish. The most important oxidation states are the trivalent and hexavalent chromium. So take out your chemistry textbooks to refresh you how that might be related. So trivalent and 3 valencies and hexavalent 6-valent chromium. But we are looking mainly at the trivalent chromium is very important. The trivalent form is the most stable and important form in biological and medical studies. Chromium has this ability to form coordination compounds and chelates. Okay, now the bioavailability of and the effects of chromium, the bioavailability of chromium depends on its characteristic ability to form coordination compounds and chelates. And the chromium bioavailability and the biological effects will depend on this chemical nature of the compound in which the chromium is present. Okay, I hope you're following that. So, chromium compounds. Organic compounds are more bioavailability in some respects, but the chromium picolinate is a very highly biologically available form, and chelated chromium and chromium amino acid complexes exist, and you can find chromium naturally brewer's yeasts and things like this, but at varying levels. This is the problem. They are at varying levels. But chromium picolinate is a stable form, and we can determine its chromium content, and it's uniform in its chromium supply. The inorganic compounds, well, the chromic oxide you will see in papers is used as a dietary marker. It's completely non-available. It's not a nutritional source of chromium. It's there for studies, for digestibility studies, but it's being replaced now with yttrium oxide and rare earth metals because chromium oxide is not a very nice thing to work with, okay? But chromium chloride is being used for fundamental studies, and it's not a reliably good source because it has a toxic margin. It is a toxicity associated with this above optimum levels, and it's very easy to get into that zone. Chromium in its active form, well, the glucose tolerance factor, GTF, is also called, well, it's basically a complex of chromium, dinicotinic acid, and glutathione complex. But the GTF, the chromium tolerance, the glucose tolerance factor, is a very important, involved in, it's in vivo, synthesized in vivo from absorbed dietary chromium, and it acts as a physiological enhancer of insulin, and it's insulin binding proteins binding to insulin, and it then increases the sensitivity and the uptake of glucose and insulin interactions by a factor of 3-fold. So it's been advocated in human clinical studies in relation to diabetes type 2 as a means of mitigating high levels of circulating glucose and to elevate the glucose insulin response. And insulin is present in fish too, but it may have a slightly different purpose, but we should be looking at the GTF tolerance factor in aquatic species as its main metabolic activity. So here we have the structure here, the chromium in the middle, and we see some of these non-essential amino acids. These may be linked to various protein structures and peptides, but we have the chromium in the middle there in this sort of balanced form interacting. These are covalent bonds which can then interact with the proteins. So this compound, these are covalent bonds here, but then we have this complex with chromium 3. So the compound contains trivalent chromium, nicotinic acid, glycine. Here's a nicotinic acid element, and glutamic acid and cysteine. Okay, so cysteine is a very important sulfur amino acid. Chromium toxicity, well, as I said, this is why people are afraid of very high levels. Chromium chloride is not nice. And we will look at that in more detail in a minute. So trivalent chromium in very large doses, no. Okay, but the role of chromium in glucose metabolism, you have these cofactors for insulin. We have chromium here. The chromium is known to increase the size of the pancreatic beta cells, which secrete insulin, and it can enhance the affinity of insulin for its receptor sites. Very interesting, especially with the rise of diabetes in the world. Protein and lipid metabolism, we know chromium plays a role in lipids, in the metabolism of lipids and lipoprotein activity. So this is very interesting. Chromium has this potential to regulate protein metabolism, and it can also affect the integrity of nuclear strands of DNA. Okay, I should put in a DNA and RNA structures can be improved because chromium may play a role in antioxidant protection of DNA. I think we can show that later. The effect of chromium generally in animal nutrition, where we know about it in humans, improving insulin metabolism in fish, we can have evidence of that. We know it improves carcass characteristics in pigs, broilers, chickens, and cattle in relation to fat content, and back fat measurements in pigs have been studied with chromium supplementation. So we know we can have some influence there on fat deposition. We can see increased body weight in horses and pigs, improved gain, improved immune responses, and the reducing of mortality, morbidity in calves, early weaning calves in dairy and beef farming. We can see also improvements in egg quality in birds and increased fecundity, production of ova and milk in pigs and in rodents. Lots of studies on rats, by the way, in laboratories around the world. So the importance in fish nutrition is we see as a source of energy. Starch is a source of energy release from compounds. Chromium is not a source of energy itself, but it can improve energy utilization. Starch is our principal energy source for most omnivorous species. So it's important to look at chromium in relation to starch metabolism and glucose metabolism from starch. And we know that the use of non-protein energy sources, such as with carbohydrates and lipids, is a means to minimize nitrogenous wastes into the environment. So we are formulating diets now more and more by sparing the protein with higher levels of non-protein energy. And that is why I think chromium is going to become more important. Some fish are carnivorous. Most of our high value species, actually, like salmon and trout and bass and bream, they have relatively, I should have to put relatively poor control over glucose levels of metabolism. The turnover of glucose in rainbow trout and salmon is roughly 10 times lower than you would find in a rat. So it's because of different enzyme activities and enzyme expressions that cause this effect in fish. The aims of the research has been to study chromium on growth and carbohydrate metabolism in the common carp, for instance. I've done quite a lot of work on the common carp and Cyprinus carpio, and to investigate the influence of chromium on body composition, liver enzymes, and blood parameters. And I've been looking at chromium on DNA. These are papers coming out from my past work with my students from Iraq. We're talking some years back now, but we had them published in top journals, and I think it's part of our chromium story. Our journey on chromium today is to look at fundamental nutrition research. Okay, so the chromium, we did an experiment where we did experiment number 1, chromium requirements. Chromium source is chromium chloride hexahydrate at different levels of concentrations in the diets. Okay, and we can see here that the range was from 0 in the control right up to 1.5 milligrams of chromium. But then we had this supra level of 2 to look at the effect of extreme levels of chromium. This is an artificial situation because we would not perhaps put that in the diet, but we want to see the effects of potential toxicity. And we can see here the study, the typical stocking densities were for experiments, 20 fish per tank in triplicate. Over a 4 weeks, they were fed a trout diet to condition them 2 times a day, followed by withdrawal of the diets, and then we put them on the experimental diets. We did this for 10 weeks, and fish were fed 3% body weight 4 times a day at a nice temperature of 25 degrees Celsius for the fish. Okay, so it's a controlled study in-house in the laboratory. And then we can see the typical diet formulations. We have starch being a major source of energy. We have a fish meal here, maybe higher than some formulations, but 25%. And we then have plant ingredients like pea protein. This is gluten, corn gluten. And we have oil, fish oil, and sunflower oils. And when you have the chromium source and the added value and the analyzed levels. These are the analyzed levels. I hope you follow that. Now, the thing is, we did a study then and followed it through. And you can see there very nicely that when we supplemented the diets with chromium, we lifted the performance. The performance was elevated. And we can see here that the control group and the very high chromium was tracking lower. We had a noticeable increase in growth within the range of optimum requirement for chromium. Okay. Okay, I hope that is okay with you. I have a bar at the top of my screen I'm trying to get rid of. So anyway, performance. You can see here very nicely that over the control, we saw an elevation in the protein efficiency ratio. That is the protein gain, the percentage, the gain in protein was higher, okay, relative to the feed intake. So we have a nice improved retention of protein, and we can see here a much better feed conversion ratio for the chromium supplementation. They start to decline when we have the high chromium. So there's some changes which are not positive. They go back to it's still as good as the control, but the chromium supplementation within the range of optimum was better. Okay, and then we can see some enzyme activities. We can see here that some of the key metabolic enzymes, such as alanine aminotransferase, which is indicating protein, the burning of protein as energy, we don't want that. But what we are seeing now is a reduction in protein being wasted as an energy source. So we see the protein transformation of the alanine aminotransferase, where we see that feeding into metabolism for energy is reduced. And we see a higher activity for carbohydrate-related enzymes, such as G6PD. We see glucose 6-phosphate dehydrogenase improve with chromium supplementation, nicely up to our optimum level of 1. Okay, and then we also see hexokinase, which is a kinase enzyme for the phosphorylation of glucose, is elevated at these levels. Okay. Maybe not synchronized, the different enzymes work differently, but the trend is there. And then when we study liver histology, we see that when we see fish fed very high levels of chromium, we start to see some negative changes in the liver architecture. Here's a normal liver. This is what we should be going for, but we see here more lipid vacuoles and droplets building up in the liver. This is indicating a suboptimal liver performance at the high level. So we have to be very careful what we do. And again, with intestinal morphology, we see very nice effect. This is a control, but we see increasing level of lamina propria, which is the underlying tissues for immunological function. We see a nicer distribution here with chromium at an optimum region of level. There's always some chromium, by the way, in the diet at the background level. But when we go to high chromium, we start to see some changes which are not good. This is a kind of clubbing effect and inflammatory response in the gut. So we are seeing at the higher levels some ultrastructural changes in the intestine that we don't really wish for. Okay, so the chromium bioavailability, well, we have all these different forms, and we did an experiment. Experiment number 2 was to look at how these different forms might work in the fish. So we had the standard, chemical form, and then we had this very nice picolinate, which is much more stable and has a higher efficacy and safety associated with it. Okay, and we tested this at the level of 0.5 milligrams of chromium per kilogram of diet. So we had the same level, and this is what we did. Here's the same kind of experiment as before, same basis, same plan, same types of fish, and from the same fish source. And then we see the diet formulation. Now that is just the background diet that we supplemented to. It's a basal diet formulation, okay? And then the chromium bioavailability. This is what you want to see. We see here the picolinate and the control. The control is here, but when we have the picolinate, we start to see we see elevation in performance, and the picolinate works very, very well. It's highly available, and it raises the profile of the fish's growth, and we can see a lifting effect. So it's a very marked, significant improvement with optimized chromium supplementation. And that confirms that the picolinate works very, very well. Okay, so we did the growth study, and here's the data. If you want to look in more detail at the metrics, we have here the initial weights of fish, we have the final weights of fish, we have the specific growth rates of the fish, and the mean SGRs of the fish. So we see here that the specific growth rate, which is the relative growth rates of the fish, is consistent over the time course. So they grow here in the control at 1.85% body weight per day, and then they become 2.1, and these were significant. And then it starts to fall back when we have the higher superdose. Okay, so everything is pointing towards an optimization around 0.51 milligram inclusion rates. And then we see here the activity, the growth of the, here we see some enzyme activity profiles like we did before. We see the hexokinase, and you can see here how very nice. This is really good. It shows that we have the highest response for the chromium picolinate. So it's more, it helps to activate this enzyme even higher, which is very good for glucose kinetics and the kinase action. This is the phosphorylation of the glucose, priming the glucose for the glycolytic pathway and energy release at the level of the cytosol in the cell, and also affecting glucose metabolism and production of ATP. So this is very interesting. So that shows very nicely when we start to use this form of chromium. And then again, we see higher activities with glucose 6-phosphate dehydrogenase. We also see it for high levels of chromium too. But we do see for chromium supplementation in general, higher activity of this enzyme, which is an enzyme associated with glucose catabolism as an energy for the energy release from glucose 6-phosphate dehydrogenase, and these are in the livers of carp. I should add, these are enzyme activities measured in the livers of the common carp, Cyprinus carpio. Now, I mentioned in the early part of my talk about DNA. DNA and RNA are very fragile, as we all know, because we're now, in terms of vaccines, we know a lot more about DNA and RNA in the news. We hear a lot about this now in the news, but we know that we have to freeze RNA at minus 80 for some of those vaccines because of their ability to break down. They degrade very easily. And that happens in vivo, in the tissues of us and animals too. So the COMET assay is a measurement of the fragility of the DNA under an electric field. It's like doing what we call electrophoresis. And we are pulling on the DNA. We're trying to pull the DNA apart. And the DNA here in the control, you can see under a certain time course, it's fuzzy. There's a little bit of a tailing effect. But when we have 2,000 ppb here of chromium chloride at the highest level of 2 milligrams per kilo, you can see really the effect of damage of chromium, okay? But here in the middle, we see very nice, especially with the chromium piccolate, we see very nice, quite pristine DNA structures. So it's more resistant. They are resistant to tearing. So only at high toxic levels do we see this, but we see an improvement against the control for both forms of chromium, but especially here. And this is a test for, we do this quite a lot for checking out on toxicity of other things too, besides metals. Okay, now here we have the liver histology, and this time we can see here that the diets fed with the chromium at 0.5 milligrams per kilo were very similar to the structure in the control. So there's no damage to the livers here. The livers are functioning properly. So this is nice evidence that, that chromium is a safe compound. Now then, of course, pathway intermediates are very interested in chromium. That's why we have this talk today. This is why we are taking this journey onwards to study chromium. And of course, we need to supply chromium as a product. There's a lot of commercial interest in this now. So we are very keen to talk today a little bit, not too much, but GROMACS. This is the trade name for their chromium source. And from fish studies, limited studies so far, but under evidence-based studies, under laboratory conditions, we are able to demonstrate in reality, in practical terms, the benefits of GROMACS commercially and for the farmer. What's important is the clients such as yourselves and how this could relate, as we said in the beginning, to enhance and improve aquaculture. It's all about improvement. And so a study with GROMACS, a trial with a Nile tilapia, a very important species around the world. I've worked a lot with tilapia and continue to do so. And tilapia, a beautiful fish, is about 8 million metric, 7 point something million metric tons of tilapia produced in places like, China, Indonesia, Vietnam, India, Pakistan, all over the world, in the UK as well we have, and mainly in the United States of America as well. And they are growing tilapia in very different conditions, in closed systems intensively or in open ponds, and such as a lot of variation in the way tilapia are produced. So a study with 400 Nile tilapia, which were fed twice a day with 2 experimental diets for 8 weeks. 2 treatments were done in 4 replications. That's a very good design with 50 fish each. So we had a control diet, a basal diet, and we diet supplemented with GROMACS to meet the chromium requirement. Okay, and with 0.05% GROMACS inclusion, a supplement in diet number 2. And the measurements was growth performance. And as I mentioned before, stress is a very important parameter, and serum cortisol level, which is an indicator of stress. And this is not published data, but you're very privileged to have a glimpse of this data. Okay, and water quality was perfect for the tilapia. It's a nice temperature. The temperature was not going above 30. This is a very nice temperature for tilapia to grow optimally. And the water quality measurements, the nitrites and the ammonia levels were well within the tolerance of tilapia, below any problems. Okay, and dissolved oxygen levels were very nice for tilapia, well within the tilapia requirements. So we see here the results of this. We see supplementation of GROMACS increased the performance. Final weight of tilapia, here we have the initial weights here, and we can see here very clearly an elevation, and this was significant through statistical applications. We could see this was a significant effect, and that is a very nice improvement. And if you look at it in terms of body weight gain and SGR, the SGR, specific growth rate, climbs from 3.67% body weight per day. That's very good for tilapia. But look what you get with this. It's a margin, a very important improvement. And so that is a consistent gain right through the growth period. Okay, and the final, you can see here, the body weight gain, the gain was 176.3 grams from the beginning. So that's a very nice improvement. And that's basic data, fundamental data. Okay, and then what was very nice here, and I've never seen this done so nicely before, this was the effect of looking at serum cortisol levels. Now, cortisol is a hormone associated with chronic stress. And you get that with fish, chronic stress. You get acute stress, short stress, and acute stress, like for instance, if there's a massive lightning bolt or thunderstorm right over the fish tank, you get an acute stress. But the cortisol reflects chronic stress, the stress that's always there at a lower level, but still it can affect performance because it takes up energy to deal with stress. So here we see a reduction with GROMAX of the stress. This is really nice. The cortisol level in the serum has dropped to a level of 23. That's quite interesting because it only takes a little bit more of this cortisol to activate stress-related parameters. So although it doesn't look, it is actually a significant reduction in stress, but we don't know what would happen if we add more chromium in this form to the diet. We should be exploring a wider range. I think we should be looking at a wider range when we have stress interactions like this. So don't forget, this was a control and a one treatment experiment. So there's much, much more to learn here. And then Pathway Intermediates took a look at the climbing perch. I'm not too familiar with this species, but I know it's becoming very important and people are interested in this. It's a great model to work with. So here we have a big experiment in a way, but more fish, 400 fish, and then they did a study with a basal diet and the chromium supplemented again, but they measured growth performance as a repeat study on a different species. And the climbing perch, of course, well, there's a lot of perch-related species. In fact, a tilapia is a kind of perch too. And we have marine perch, we have perch in the ocean, and we have a lot of interest in perch-like families of fish across the world. So this is a great model to have in our pocket. Okay, so the fish were fed to satiation twice a day. That's quite important to state. They were not fed as a percent of their body weight. They were fed to satiation. So the fish were able to perform without any inhibition of appetite suppression or anything like that. They were fed to satiation. And the water quality parameters, I won't say too much, but they met the demands, so they were well within the tolerance range of fish. And it's important to get that right. However, I should say, sometimes it's good and interesting to work in suboptimal water quality, because if everything is perfect, then we don't see some of the attributes of our products. The potential improvements are sometimes better understood in conditions which may not always be the best, okay? Because we have more stress when the water quality is suboptimal. So we must be looking at criteria where perhaps elevated temperatures or lower temperatures, and this could be very important in shrimp. We must be looking into the future of how we might use this for shrimp, which are very prone to stress, okay, and subjected to lots of different water conditions. We'll come back to that point. And the growth performance, they did a study, was very interesting, because it showed clearly again, right, we saw differences in other parameters. Now, they added another interesting factor here. They looked at fat reduction in the fish. Now, the fat in the fish, sadly, it's like with me, the fat goes to your belly. It goes into your viscera. It goes into the gut. And when we process the fish and harvest the fish, we don't want to throw away all that energy in fat in the gut. We want the fat to be used to grow the fish. So this was interesting because we saw a reduction in the fat to gut weight. Maybe I should be taking GROMAX. It would help me slim. So anyway, we see a reduction here in the fat visceral content. And it is very significant, as you can see from the superscript here. And then when you look at survival, this is really interesting. Survival is important because survival is key thing in aquaculture. And for a farmer to have a survival of 98% compared to 93% is significant, very economically significant. Okay, if we translate that into economics, which is something we're going to show, that is very important. The economic gain relative to diet costs. If you factorize in the diet costs, then you will see advantages. And I thank Young Kang for doing some interesting scenarios. This is an economic analysis model. It's a model. It's an example. It's an example in US dollars of what you would get in terms of income, overall feed costs. And this has been done for 100,000 head as a model. You can translate this into tons. You can translate this into kilograms, but it's 100,000 per head fish model. The overall feed cost for that would be here in US dollars over 11,539 US dollars, profit margin USD, and then the return of income. And we can see here with added GrowMax, you get a nice gain per 100,000 head. It's no units there because it's a relative term. Okay, and you can see then the differences and profit differences. So it'll be very interesting for us to apply that to different scenarios, to different species of fish. Now, we've been looking a lot at warm water species and examples there like carp and tilapias, but you know, I would very much like to see chromium research done on colder water species like salmonids, even though we can perhaps get them to improve their utilization of carbohydrates. And when I said that there's relatively differences in carnivorous fish, they are able to utilize some carbohydrate over time. They can actually adapt to slightly higher levels than maybe normal, but we should be looking at chromium in relation to carbohydrate efficiency generally, and in general, other factors that would be beneficial for chromium. But fish do use glucose, but they manufacture their glucose. They make glucose through a process called gluconeogenesis. So we need to perhaps better understand this relationship of trace elements in energy utilization in general. And for carnivorous fish, insulin, which is this insulin factor, which is chromium dependent, may improve protein utilization as we see in the carp. So it's not exclusively a carbohydrate issue. So I think we have some interesting challenges lying ahead. Now, you might want to go into this in more detail. I hope you do. You want to study chromium? Well, I think there's quite a lot of references developing here. Some of my work with Arafat is my Iraqi student. She's from Basra in Iraq, and she came over to, after the tragedy of the Gulf War and everything, they came over from Iraq to study, and they wanted to improve their academic standing. So we had grants and funding from the World Bank to support them. So I want to thank them for that. And then we see the NRC requirements is something you should be looking at. So with that, my take-home message is chromium is an important trace element and can definitely enhance aquaculture performance. Thank you very much.